Much, Joni, for that wonderful introduction and opening. It's my pleasure now to introduce Passley Hargrove. She's going to be bringing us into today's first session with the TraCE-MPS grant sites as well as the botulinum toxin patch programs. So, Passley.
Well, thank you so much, Nick. Thank you all for being here today, and thank you so much for the Critical Path Institute for being such incredible partners in the creation and development of this meeting, and we hope you get a lot out of it today. So I'm just going to briefly introduce the NIH Tissue Chip for Drug Screening program. We started about 2012 with the program goal of developing human-based in vitro 3D culture systems, tissue chips, microphysiological systems, organs on chips that emulate organ physiology and function. And really the goal here was to bring in a diverse set of scientists, those who had expertise in stem cell biology, microfluidics, bioengineering, to look at risk assessment, not only for looking at safety and toxicity of promising therapeutics, but also efficacy analysis. This is just some of the organ on chips that we have looked at throughout the program. And so we really started in 2012. All these programs are these initiatives are about 5 years. We started with the low-hanging fruit, which was safety pharmacology, to really assess toxicity where no physiological and pharmacologically relevant models were available. 5 years later, we pushed into disease modeling and efficacy testing for all types of diseases, rare, complex, Mendelian, non-Mendelian, to capture the pathophysiology, mutation spectrum, and phenotypic diversity of human diseases and conditions. During that time, we also worked with 2 main tissue chip testing centers, as well as the MPS database, to build confidence in the technology, look at the robustness, reproducibility, and reliability of the technology that we could onboard from our developers. About a few years later, we pushed into the precision medicine space, looking at can we use tissue chips to inform clinical trial design, planning, and execution, and to stratify patients based on response or lack of response to a candidate therapeutic. Recently, we focused on building community. So for the first 3 years, we funded the MPS World Summit, and they're now completely viable on their own, and they have also spun out an international MPS society. And really what we're focused on today is pushing towards regulatory acceptance. We've partnered with the FDA since the inception of the program in 2012, but this is really our first kind of stride to, with our translational centers for MPS and our botulinum toxin potency assay teams, to really kind of propel us to the next avenue. And so we also have the Tissue Chips in Space 2.0 program, which Joni mentioned, and Dimitri will talk about in the next session. So our TraCE-MPS centers were started about 3 years ago, really with the main goal of accelerating the translational use of microphysiological systems in drug development, pushing for regulatory acceptance and adoption for industrial use. This is a letter of agreement between NCATS as well as the FDA and the Critical Path Institute, where we're focused on qualifying MPS as bona fide drug development tools that are fit for purpose for industry and have highly specific contexts of use for regulatory qualification. We have 4 teams that will be presenting today. We have Jonathan Himmelfarb and Bino Friedman's team from Mount Sinai University of Washington. We have Yvonne Rusin team and Ron Minan's team from University of Texas Medical Branch, as well as Texas A&M. We have the Taylor team from University of Pittsburgh, as well as the McGrath team from University of Rochester and Duke University. So we have been working, these teams have been working for submission to the Innovative Science and Technology Approaches for New Drugs program, or ISTAND program. This program was started in 2020 and became permanent about 5 years later at the FDA, and really accepts submissions for qualifications of drug development tools that are currently out of scope for existing programs. I did want to highlight the letters of intent that have been accepted from our teams through this program. You'll have access to these slides, and you can actually click in and you can look at all the feedback from the FDA through these hyperlinks. But I did want to point out that 25%, or at least 1/4 of the accepted letters of intent, are from our TraCE-MPS awardees and our bot patch awardees. And so we're very excited about where this is moving. And I'm also excited to talk to you today, and we'll have 2 teams from the botulinum toxin potency assay. This is also a letter of agreement that was specific to this program, looking at development, regulatory qualification, and commercialization of alternative NAMs that specifically utilize the neuromuscular junction platforms to replace, as they said, the LD50 or mouse lethality bioassay for botulinum toxin. We have two teams from Curie Bio looking at high throughput NMJ assay for Botox screening, as well as Jay Hickman's team looking at, from Hesperos, looking at human neuromuscular junction platform to evaluate botulinum toxin potency. So these letters of intent have also been submitted to the FDA ISTAND program. And with that, I just want to thank everyone here for just your incredible work throughout this program.